All right, welcome back. I'm gonna let that play just a little bit in the background because I love that song and I love that movie, Meet the Robinsons. If you haven't seen it, it's definitely a kids' movie. It's so cute though, and the reason I'm playing it is because cells are little wonders, aren't they? And try to sort of match these songs to match what we're talking about, which is actually hard to do. I probably do more time wasting time probably doing that. But anyway, so we're going to talk about some cells. Let me bring it out so we can say a little quick hello. I'm having a problem with this today. Let's see if I could get this back. Okay, bear with me. Bear with me. Oh, there we go. There I am. Okay, I'm having some technical difficulties, so bear with me here. Okay, welcome back. This is Dr. DeChavo. This is going to be the third part of your, bear with me, third part of your chapter 3. So this little section is the last little bit of notes that we'll have for chapter 3. And in chapter 3, we have talked about the cells, the different parts of the cells, how they interact, how the cells have different small, what are called organelles, which are just like organs that we have in our body. And now we're going to move into a little section about DNA and how DNA replicates, which I actually went over in chapter 2. But I'm going to do a brief little review of that. And then how cells undergo mitosis, okay, which is their replication, but the steps of that mitosis. Okay, so cells, the smallest living units. I'm going to bring you back in on this. Hopefully we don't have more technical difficulties. If we do, we'll work around it. Okay, so summing up protein synthesis. I'm going to walk you through this slide, then I'm going to pull you out so you can look at what I'm doing. Okay, so now remember, proteins are comprised of amino acids. So we eat proteins. You know, I left turkey burgers at my house tonight for my kids and my husband. I didn't actually cook it though, so I can't take claim for that. They will eat those turkey burgers, and those turkey burgers have in the burger itself a lot of protein. Our mastication or chewing, our stomach acids, and our digestive processes break up those turkey burgers into amino acids. Our digestive system absorb the amino acids, and then our body strings those amino acids together to form eventually a protein. Okay, it's called a polypeptide because there's multiple what are called peptide bonds. If you don't remember this, review chapter 2. Okay, there are peptide bonds between those amino acids. Long chains of amino acids form a protein. Okay, the way that we got this sequence of proteins is from the DNA. Okay, so I'm going to pull you back out so we can talk about that briefly. Hopefully this works right this time. Yay. Okay, so what happens, and if you don't remember DNA structure, review it please from chapter 2, is that alpha helix structure of the DNA separates. Now remember, the strings of DNA are long, 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 long. And in our body, in those strings of DNA, we have what is called the genetic code. And along that genetic code is the sequence to create the strings of proteins that build the structures of our body, our hormones, our enzymes, etc. Okay? So somewhere along this long chain of DNA is the sequence that we need to make a specific protein. So the whole DNA doesn't create it, just this little sequence of it, okay? Your book goes way more into this than I'm going to go into it. I'm just going to go into the brief explanation of it, okay? So if we're looking at one little section of that long DNA chain, we've got nitrogenous bases or nucleotides, adenine, guanine, cytosine, and thymine in DNA. Okay. In DNA, it will have its complementary pair. Okay, so we've got that alpha helix, the nitrogenous bases or nucleotides, and they're always paired, as we should remember, okay, this way. This is DNA. So if we want to create a protein, the protein that we need to create comes from this segment of our DNA. What will happen? I'm going through this quickly because we've already gone through it. is it will split, okay? And what will happen if this is the sequence that will eventually create the protein, mRNA comes in and creates a complement to this. Okay, so mRNA comes in, okay, will form a complement. Don't forget there's not T, there's not thymine in RNA. It's going to be uracil, guanine, cytosine, and adenine. Okay, so now this sequence, which is mRNA, is carried out of the nucleus and into the cytoplasm. Okay, it will go to the endoplasmic reticulum, and the ribosomes on the endoplasmic reticulum is where this mRNA is going to. Okay, and through a series of very complicated steps, that mRNA will give the sequence to the ribosomes, and the ribosomes will actually start to string amino acids together. Okay, because keep in mind, I'm just giving you a segment here. If we're actually creating a protein, we'll send out a longer segment of these nitrogenous bases to give the template for these amino acids. Okay, so the way that it does it, I'm going to put you back to the notes now that you sort of have this visual. The way that it does it is 3 of those nitrogenous bases or nucleotides or nucleotide bases, all interchangeable references. And I switch them because you'll read different things in different areas. Okay, so you get used to hearing that. So 3 of those nucleotide bases form what's called a triplet. This triplet, so those 3 nitrogenous bases, is the code for an amino acid. Okay, I'll say it again. 3 of those nucleotide bases is the code. It's also known as a triplet. This triplet is a code for 1 amino acid. There's a whole list of triplets and what amino acids they represent in your book. Don't worry about it. I want you to understand that concept. Okay, so those nucleotide bases will be carried out with the code on the mRNA out to the endoplasmic reticulum, to those ribosomes, okay? And they will start to form these triplets, or they do form the triplets, which codes for the amino acids, and you start to get these long chains of amino acids, okay? Oof, it's tough. If you have questions about that, listen to this part again. If you still have questions, come see me, okay? So, transcription is going to be that transferring of the sequence from DNA to RNA, okay? mRNA is messenger RNA. It comes in, it gets the sequence for those nitrogenous bases or nucleotides from DNA, and then it goes back out into the cytoplasm, you can see there, and it undergoes a process called translation, in which that mRNA gives that sequence for those amino acids, forming the polypeptide. Okay? So transcription is going to be in the nucleus. Translation will happen out in the cytoplasm, creating the amino acid chains, I should say, forming polypeptides. Okay, how do cells replicate? This probably sounds a little bit familiar. We're going to be talking about mitosis here. We're talking about meiosis when we talk about reproduction at the end of our journey in the spring. Okay, so the cell cycle is comprised of 2 major groupings. There's interphase, during which a cell is resting and also replicating its DNA. So in order to form 2 identical cells, so I wear off a whole bunch of my skin cells, I've got to create more. Those cells have to undergo what's called interphase, where they will replicate the DNA. So that way we can create the same chromosome numbers in future pairs or in future cells. Okay, so we duplicate that DNA, we divide the cell, then we have 2 identical cells. Okay, cell division itself is mitosis. So interphase is resting and replication of DNA. Cell division, in which the cell and the nucleus are split, is called mitosis. Meiosis may sound familiar. That is the replication of reproductive cells, sperm and eggs. Okay, so cell division, mitosis. Don't worry about the G phase and the S phase, okay? We're just going to talk about mitosis and the different parts of it. Essential for body growth, right? If we don't have mitosis, we cannot repair tissues. Now, there are some tissues that can't be repaired: nervous tissue, skeletal muscle, and cardiac muscle. If you damage those tissues, you will not create new cells. What will happen actually is you'll create scar tissue that will fill in that area. Okay? Chromatin. So we're thinking now about the nucleus. Okay? We're thinking about the nucleus. Chromatin is long strings of DNA, and DNA is actually organized into what are called nucleosomes. Okay? I have a good picture on the next page if you want to look at it while I'm talking to you. So there are these 8 disc-like structures called histones. They're proteins. They're little discs that are structured very sequentially. The DNA wraps around it, and it helps to coil the DNA a little bit to keep it from getting too bunched up. Okay? So chromatin is when the DNA is stretched out. It's before it starts to replicate, and it actually looks like a bowl of spaghetti. Okay? As the cell starts to get ready to divide, the DNA will replicate and then form chromosomes. So when you're looking at chromosomes, it's when it is replicated right before division that chromatin condenses, forms those X-like structures. Okay, 1/2 is called a chromatid. I'll show you a picture in just a second. And it's connected by what's called a centromere. So it's almost like if we look at this picture, first of all, you can see that super coiled DNA. It's wrapped around those disc-like histones. They're proteins, groupings of 8 proteins, and it helps to just organize it, okay? And then it's wound and wound and wound and wound and wound, and you can see how much DNA is condensed down into that chromosome, okay? You could see the arrows pointing to the chromatid, okay? And then there's an identical chromatid on the other side. That's the replicated part. The centromere would be like the center of a bow tie in the center of that chromosome. That helps to hold it together, okay? So, cell life cycle. Interphase, I'm waiting, and cell division, let's split. Okay, so interphase is it's waiting, it's resting, it's duplicating the DNA. Mitosis is when you're actually going to split those. Okay, and here we go. So interphase, resting from dividing. You can read through all that. What's what we've gone through. DNA replication occurs. Those nucleosomes unwind. DNA separates. It duplicates itself. Okay, and then rebonds actually into those new chromatin strands and chromosomes. So what happens, and I've sort of explained this already as well, but I'd like to give you a little visual. Okay, with DNA, what happens? Okay, so forget RNA. We are not talking about RNA now. We are just talking about DNA. We're talking about DNA as it starts to get ready to form 2 identical cells. That long strand of DNA, okay, will split. And if you can imagine, and once again, I went through this in chapter 2, so if you need a little review, here it is. And what will happen, okay, so here's the split strand. What will happen is it duplicates itself. And by duplicating itself, you form 2 identical strands with the same identical genetic information. So you get 2 identical cells. It's pretty cool. Okay? So that full length of DNA, that long strand, will separate and will replicate. Okay? So, the interphase. So that happens during interphase. Okay? Now, moving back into the picture, I have a nice little picture there. It actually, along the full length, because it's so long, if it were to start at one end and unzip the whole length, it would take forever. So there's actually what are called replication bubbles that happen along the full length of DNA when it's ready to split. they start to separate, they start to replicate, you get two identical cells. Pretty cool. Okay, here's a picture of a chromosome. You can see a chromatid is going to actually, on this one, you can have a short arm and a long arm. I wanted to show this to you. So if you were to divide this with a vertical line up and down, okay, the half on the left and the half on the right would be identical to each other, okay? And they're held together by that centromere in the middle. You can read through this on your own. I think the numbers are astounding. Really, I sounded a little Australian there, didn't I? Astounding. Okay, so pretty cool. Cool stuff. And karyotype. I think this probably looks familiar. Okay, karyotype is going to determine people's genetic material. It's taken right before the cell starts to split. So you have the full complement of those 23 pairs, okay, or 46 chromosomes. And you can see with the karyotype if there's genetic deformities with someone. Okay, so once again, I won't test you on this. I put this down to explain to you how you take a karyotype. And cell division, so mitosis. So mitosis is when the division of the nucleus begins, and cytokinesis is division of the cytoplasm towards the end. I have a nice little video here for you. I'm going to back it up so you can hear that beginning.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
Okay, so amazing, the information that's out there anymore. Okay, so brief little explanation of mitosis. Now, I had difficulty when I was going through school trying to remember all the phases of mitosis, so I've given you silly little sayings to try to help you with this, okay? So there's prophase, metaphase, anaphase, telophase. Prophase, I nicknamed the prochromo, okay? What's happening is chromosomes are starting to form. Centrioles are little structures that help to sort of solidify, what's the word I'm looking at? Anchor. Okay, they help anchor what's called the mitotic spindle, which is a series of myofilaments or filaments or microtubules, I'm sorry, which start to line up the chromosomes so they're ready to split in half properly. The cell then moves into what's called metaphase. Okay, me on the equator, so me being chromosomes. The chromosomes line up on the equator, okay? We've got those mitotic spindle fibers attached to it, ready to pull them and split them in half. And they split. The chromosomes split during anaphase. They're pulled to opposite sides of the cell, okay? You then have that nuclear envelope, nuclear membrane, start to form around it. And now you have two copies of the same genetic material. The cell then moves into telophase, tell the cell to split. Now it's time for the cell to split. It forms a membrane around both, okay? And that process is called cytokinesis, when the cytoplasm splits and now you have a nice cell membrane around both cells. You have two identical sister cells. Pretty cool. Okay. Developmental aspects of cells. So just some things to keep in mind, actually some definitions I should really say about cellular events. Okay, stem cells are undeveloped cells that we find in embryos, but we also find them in different parts of our body. Okay, stem cells are undifferentiated cells that when placed next to other cells will start to turn into those cells. So you can see how stem cells would be an advantageous developmental stage to be in. If we needed new cells in the heart, we could place stem cells in there and they would turn into heart cells. Okay, it's a matter of getting the right stem cells what's the word I'm looking for? Taken, okay, from a specimen. We could actually get stem cells from our bodies at this point and keeping them viable so we can plant them so they can start to propagate. There's a whole series of complicated steps to that. Now, cell differentiation is when undifferentiated cells start to turn into specific types of cells. Cell differentiation really is applicable when we're talking about embryological development. Okay, embryos start as a cell, then they start to turn into 3 layers of cells, and then they start to differentiate into the different types of organs and tissues. Okay, so cell differentiation is when a cell gets turned on and it's decided to turn into a specific type of cell. It's pretty cool. Apoptosis is programmed cell death. It is when a cell becomes malformed or dysfunctional. The body will sense it, that cell will sense it, and it will actually kill itself. This is part of where cancer goes wrong. Cancer will not undergo apoptosis. Those cells keep differentiating even though they are abnormal, and we don't know why. That's why we can't cure cancer yet. Hyperplasia is an increased production of cells. So if you were to think about a callus, that would be a great example. That area where the callus is going to be an overproduction of cells in response to some sort of irritation, such as bad fitting shoes. Okay, so hyperplasia. Atrophy is when cells shrink. Atrophy happens, especially post-cast. If somebody has a cast on, you can see that their leg gets quite a bit smaller even after 6 weeks. That's because those cells in the skeletal muscles are not used, and they will actually start to shrink. They become less metabolic, so they don't need as much energy. Okay, so, and that can actually be reversed once the cast is taken off. Okay, so atrophy is when cells shrink. Okay. I put this in if this helps you with some visuals as to the different phases. I'll test you on what we spoke about though. I won't test you off these slides. Okay, but different people need different things to help understand the material. Okay, so that was just really short, little explanation. And if you have any questions about that, by all means, let me know. It's probably one of the toughest chapters that we go through in chapter 2. So please, by all means, if you have any questions, let me know. And that being said, I'm going to let you get back to Little Wonders again by Rob Thomas for a few minutes while I'm signing off. Thanks so much. Let me know if you need anything. Take care.